My name is Andrea Kasinski. I'm an associate professor in the Department of Biological Sciences and deputy director of the Purdue Institute of Cancer Research at Purdue University. So RNA is thought of as an intermediate molecule between DNA, which is the information that's encoded from our cells, and proteins. So it's usually thought of as this intermediate. So DNA will get converted into RNA, and then RNA is used to make proteins. So we think of microRNAs as really kind of the brakes. They prevent other things from being expressed. So when you lose one of these microRNAs, you can imagine that all these other things that shouldn't be around are now expressed at very high levels. And if these other things are disease-causing, you've just lost the brakes on that system. And now everything is kind of haywire. And so the work we're doing in my laboratory is to try to restore these small RNAs. And we can do that because they are small, they're easy to work with. They're not subject to all these other concerns that you have to think about with some of these larger RNAs. So these small RNAs, their role is to negatively regulate these bigger RNAs from being able to be translated into a protein product. So when you lose one of these small RNAs, many of these other bigger RNAs are now translated into proteins, and those proteins drive cancer and a variety of other diseases. So through restoring some of these small RNAs, we can kind of revert that process back to the normal situation, preventing these protein products from being made and reducing their levels so that they can no longer promote disease. We know that cancer as a whole is driven by so many different genes. So if a single microRNA can modulate all those genes, bring them down collectively, we have this enormous ability to overcome many of the things that cancer becomes addicted to.